All right, my name is Mark Kramer. I'm an associate professor in mathematics and statistics here at BU. And I'm going to tell you a bit about, a very brief amount about the data analysis and modeling we do in our group and how we attempt to make these models and ideas accessible to practicing neuroscientists. So as you've started to see and as you'll continue to see, there's lots of exciting data being collected here, and that data motivates the questions that we ask. And to give you a sense of those questions, here are a couple of data analysis questions. You might imagine you have many sensors on a brain surface. They each record voltage or some signal as a function of time. And you'd like to look at those signals and determine, are they interrelated in some way? There are lots of ways to answer that question. You might apply a measure like Granger causality, where you attempt to model the activity at one sensor as a function of its past and the past of other sensors. It's a nice idea, but for a high-density recording with lots of sensors, it's computationally intractable. There are just too many parameters to estimate. So what could you do? You could give up, or you could talk to a talented graduate student like we have here, Elizabeth Spencer, who can develop a statistically principled way of assessing this Granger causality for high-density recordings. Another question that we investigate, how do brain rhythms interact? And as a simple cartoon example, you might imagine that you have two rhythms. So you have a low-frequency rhythm shown here in blue, and then a high-frequency rhythm shown in red. You might notice that at a particular phase of this low-frequency rhythm, there's a blast of high-frequency activity. So we'd like tools that can assess these relationships, and again, they can be developed by talented graduate students we have here at BU, Jessica Nadalin, who's developed a statistically principled way of assessing these relationships. And then ideally, you'd like to detect these relationships in real time and then apply neurostimulation to modulate them. So that's ongoing work by a postdoctoral researcher in our group, Ani Woodier. And as one other example of interacting brain rhythms, here's a clinically relevant example. This blue activity shown here is an interrectal discharge, and it can be recorded on the scalp surface, this large amplitude signal. And if you look closely at this, you might notice a high frequency wiggle on top of it. These high frequency oscillations or ripples are associated with epilepsy, so you'd like to find them in patients. But the standard analysis of this is visual inspection, which is completely intractable. It just takes too much time for a clinician to try to identify these features. So what could you do? You could give up, or again, you could talk to a talented graduate student like Jessica Nadalin, who can apply machine learning techniques to make this biomarker tractable and detectable and clinically useful. So that's one kind of area that we look at. Another area is developing mathematical models, typically to try to identify candidate mechanisms that support observed activity. So you might observe interesting spatiotemporal dynamics on a two-dimensional surface recorded from a brain and wonder what are the mechanisms that support it. To address that, we can develop models that consist of interacting populations of cells that interact with each other and the extracellular environment. This is ongoing work in our group, and it's continuing again with talented graduate students and postdoctoral researchers that we have here at BU and at MGH. And then finally, ideally, we try to make these tools accessible. So I work closely with Ori Eden, he's a statistician in our department, and we try to put all the tools we develop online for others to use. We're also interested in educational material. Currently, we are doing that in MATLAB, but it's now being ported to Python, again, by talented students that we have here. And all of this follows from the data that's being collected. And so we let all of you do the hard work of collecting the data, and then we try to develop data analysis methods and models to understand them. And we're always happy to chat more with experimentalists. All right, thank you.